Welcome to this episode of Lectures with Liddington. Last episode, or last unit, let's say, we talked about the cell cycle and how cells go through these stages in their lives. And one of those points in time, we had to replicate the DNA before we could move it to different cells. All right, so DNA, cool. We've been talking about it. We've brought it up. But what is it? Let's get a better understanding of what DNA is. So for this episode, we're going to specifically talk about the structure of DNA and what it looks like. So DNA, it's an abbreviation because the real word is super long. It stands for deoxyribonucleic acid. DNA. And the reason we call it deoxyribonucleic acid is because here, this is our backbone, and the sugar that's in that sugar phosphate backbone is called deoxyribose. Deoxyribose sugar. So the shape of DNA is what we call a double helix, and this is sort of like a twisted ladder. So it's legitimately as if you took a ladder, grabbed the top, grabbed the bottom, and twisted it around. On the outside here, the handles, we have a backbone that's alternating sugars and phosphates. And then the rungs on the ladder here, those are what we call bases or base pairs. So there's one base coming from the one side, and then there's another base coming off of the other, and they pair together very specifically. So go way back into the archives, right? When we talked about macromolecules, DNA, deoxyribonucleic acid, must be made of what kind of macromolecule? Yeah, nucleic acids. And if we go back and think about that, DNA is one of our examples of a polynucleotide, right? So it is a macromolecule made up of many nucleotides. So the monomer, the little pieces that we add on together to make that DNA, that monomer of DNA is called a nucleotide. So let's look at what that looks like. So each nucleotide is composed of 3 different pieces. They all have 1, 2, 3. Here's our 3 pieces. This is a phosphate group. That is a term that you should be familiar with, right, as one of our functional groups. We talked about it when we talked about nucleic acids, and we also talked about it when we talked about ATP, right? That's our phosphate group. And then right here, is a 5-carbon sugar that makes up that sugar phosphate backbone that we just talked about. And then here, this is what makes up the rungs of the ladder. So this piece here, the phosphate and the sugar, this will make up that backbone. And here, this is a nitrogenous base. And you'll have this nucleotide will be attached to another nucleotide that goes this direction. So you'll have 2 different bases attached that make the rungs of the ladder. And then over here on the sides, we'll have our backbone made up of alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down. So that nitrogenous base, there are a couple of different options that we could have, specifically 4 different bases that we could have. And those 4 bases are thymine, adenine, cytosine, and guanine. And what you'll notice is that these bonds here, There's 2 between adenine and thymine, and then there's 1, 2, 3 between cytosine and guanine. And that is very specific. You would not be able to have any changes here because of that bonding pattern. Because you have 2 bonds here between the adenine and thymine, you couldn't have an adenine and a guanine because you have 3 bonding placements in your guanine. So why do we want to say adenine, thymine all the time? Well, we don't. We just abbreviate with letters. So adenines are A's, thymines are T's, cytosines are C's, and guanines are G. So we always have consistent base pairing, A to T and C to G. Some people will remember it as cars in the garage, apples on trees. I like to remember it as curvy letters cuddle, right? So your C and your G are both sort of curvy letters, so they'll cuddle with each other, right? So they're bonding. So this is always the case, A to T, C to G. Notice, I'll point out here, the pieces of our nucleotide. So this whole thing right here is my nucleotide. Here's my phosphate. Here is my deoxyribose sugar, right? Because it's DNA, it's deoxyribose. And then here is my base, in this case, thymine or T. So that's really cool and all, the structure of DNA, but why? What does it do? Well, we've talked about it so far in the past that DNA is responsible for storing and transmitting genetic information. And how it does this is based off of the pattern of those bases, the patterns of the nucleotides. So AATC means one thing, but TTAC means something else. So the information is stored in that pattern. So that information is stored in the pattern of bases. Think of it sort of like words, right? How you arrange the letters will give you different words, which gives you different information. And that's just how it works with DNA. So what we do is we have a pattern of those bases, and every 3 bases will code for a particular amino acid. Remember back to macromolecules. Amino acids are the monomers of, that's right, proteins. So we take proteins, or we take amino acids rather, we take those amino acids, we put them into a sequence, and the sequence of those amino acids codes for a protein. And it's these proteins that then determine the traits of an organism. How tall it is, what color it is, hair color, eye color, whether or not you have a hitchhiker's thumb, whether or not you have a widow's peak, things like that are all determined based off of the proteins that are made because of the coding in your DNA. So let's pause here and do a little practice.